Hello everyone and welcome. So today we are going to discuss about antibodies. This is our third lecture, and in this discussion we are going to look at the definition of antibody, the structure of antibodies, various classes of antibodies, important functions of antibodies, the concept of polyclonal and monoclonal antibodies, and towards the end we shall look at isotypes, allotypes, and idiotypes. So what are antibodies? So basically antibodies are globulin proteins that react with the antigens which stimulate their production. So we can also define them as protein substances produced as a result of antigenic stimulation. And it is important for you to know that antibodies account for about 25% of plasma proteins. And according to electrophoretic migration rate of blood, it contains three types of globulins, that is the alpha globulins, the beta globulins, and the gamma globulins. And antibodies form part of the gamma globulins. So next we look at the structure of immunoglobulins. Antibodies are glycoproteins and consist of light and heavy polypeptide chains. So light chains, which are abbreviated as L, have a molecular weight of about 25,000, whereas for the heavy chains have a molecular weight of about 50 to 70,000. So, and for the simplest antibody molecule, it will consist of 4 polypeptide chains and it will appear Y-shaped. So out of the 4 polypeptide chains, 2 of them will be heavy chains and 2 of them will be light chains, giving a molecular formula of L2H2. And an individual antibody will always contain identical heavy chains and identical light chains. So from this simple representation of an immunoglobulin molecule, so we can see this is where we are having the light chain. So this is the light, the 2 light chains, and these are the 2 heavy chain. So it's important for you to know that. So the light and heavy chains are subdivided into variable and constant regions. So the regions are made up of 3-dimensionally folded repeating segments called the domains. So each domain will contain approximately 110 amino acids. And a light chain will consist of 1 variable domain and 1 constant domain, whereas most of the heavy chains will contain 1 variable and 3 constant domains. For example, immunoglobulin G, D, and A will have 3 constant domains in their heavy chains, but for immunoglobulin M and E, they will have 4 constant domains in their heavy chains, and because they have 4 constant domains, so they will appear much longer and heavier. So looking at this simple immunoglobulin molecule again, so you can see here we are having the variable region of the light chain. We are having one there and another one here. So these are the variable regions of the light chain. Then again we have the variable region of the heavy chain. So they are here. This is where we have the variable region of the heavy chain, and you can also see the constant region of the light chain. So this is the constant region of the light chain, and we have another one here. Then you can also see the constant region of the heavy chain, which are, for this immunoglobulin, there are 3. So you are having one here, another one here, and another one here. So it's important for you to know that. And towards the amino terminal end, that is where we have the variable region, and towards the carboxyl terminal end, that is where we have the constant region. So it's also very important for you to understand that. Now, moving on, what are the functions of the variable and the constant regions? So for both heavy and light chain, the variable region will provide antigen binding site. So that one is very important for you to understand. And it is also key for you to know that antigen-antibody interaction involves electrostatic and van der Waals forces, and also hydrogen and hydrophobic bonds. Moving on to the functions of the constant regions. So for the heavy chain, the constant region will participate in complement activation and also binding to cell surface receptors. Whereas for the functions of the constant region of the light chain, it is uncertain or unknown. We also have other regions called hypervariable regions within an immunoglobulin molecule. So these are regions found on both the heavy and the light chains and have three extremely variable amino acid sequences at the aminal terminal end of their variable regions that form the antigen binding sites. And hypervariable regions will always contain the amino acids, about 5 to 10 of them. And like we have already said, they provide the region for antigen binding. Now, why are they called variable regions? Why are they called variable regions? So they are called variable regions because of their variation in the amino acid sequence between the immunoglobulin synthesized by different B lymphocytes. So that one is very important for you to know. So considering this immunoglobulin molecule, so we can see we are having the hypervariable regions on both the heavy and the light chain. So for the heavy chain, this is where we have the hypervariable regions, whereas for the light chain, this is where we have the hypervariable regions. And we have said the hypervariable regions will contain 5 to 10 amino acids. So you can see the arrangement of the amino acids both on the hypervariable region of the heavy and the hypervariable region of the light. So you can see the amino acid arrangement there. And like we have said, these hypervariable regions will provide the site for the antigen binding. So you can see they provide the site where the antigen comes and then binds on the immunoglobulin molecule. So we are going to discuss the light and the heavy chains. So we will start with the light chains. So we have two types of the light chains, and these two types are achieved according to the amino acid differences in their constant regions. So we have the kappa and the lambda. So kappa light chains are encoded on chromosome number 2, whereas lambda light chains are encoded on chromosome number 22. And both types can be found in all classes of immunoglobulins, but it is important for you to know that any one immunoglobulin molecule will only contain one type of the light chain. Moving on to the heavy chains. So heavy chains are distinct for each of the 5 classes of the immunoglobulin. So each immunoglobulin will have its own heavy chain. For example, for immunoglobulin G, it will have the gamma. Immunoglobulin A, it will have alpha. Immunoglobulin M, it will have mu. Immunoglobulin E, it will have epsilon. And immunoglobulin D, it will have the delta. But all the 5 classes, or all the 5 types of the heavy chains are encoded on chromosome number 14. So from this diagram, we can see that for the light chain, we are going to have kappa and kappa. For example, here we are having kappa and the lambda. So these are the kappa and the lambda light chains. Whereas when we consider the heavy chain, you will see we have 5 of them. So you can see 5 of them all the way from alpha, delta, epsilon, gamma, and mu. You can see them there, and they are located on chromosome number 14. So considering this structure of the immunoglobulin molecule, so you can also see that the light and the heavy chains are normally joined together at the hinge region. So this is the hinge region where we are having the joining of the light and heavy chains. And what facilitates or what brings about the joining of those chains is basically the hydrogen bonding, or you can talk of disulfide bonding. Now, what happens suppose you treat an immunoglobulin with a proteolytic enzyme, for example, papain. So if you treat an immunoglobulin molecule with a proteolytic enzyme, for example, papain, so you will cleave the hinge region and then break the bond. So once the bond on the hinge region are broken, so it produces two identical Fab fragments and one FC fragment. And it's important for you have a look at that. For example, in this immunoglobulin molecule, you can see that this is where we are having the Fab region, and this is where we're having the FC fragment. And we are saying you will have two Fab fragments. So this is one of it. So we are having one and two there. But when it comes to the FC fragment, we are only going to have one of it. So it's important for you to understand that. Now, what is the function of the Fab fragment and the FC fragment? So the Fab fragment is the one that carries the antigen binding site. So antigen binding sites are carried on the Fab fragment because they contain the hypervariable regions. Whereas for the FC fragment, it is involved in complement fixation, attachment to various cells, and also placental transfer, among other functions. Now, how are immunoglobulins produced? So recall that we said the B lymphocytes are the ones that are involved in production of antibodies. But we said B lymphocytes, under the activation of interleukin 4 and interleukin 5, they undergo transformation to produce the plasma cells. And the plasma cells also give us the antibodies. So in other words, B lymphocytes and plasma cells take part in the synthesis of antibodies. But the B cells will only produce small amounts of immunoglobulins that are incorporated into the cell membranes, whereas the plasma cells are the ones specialized to secrete large amounts of immunoglobulins. So the heavy and the light chains are synthesized in separate polyribosomes of the plasma cell, and an immunoglobulin molecule will contain one heavy and light chain forming a HL hemimolecule, which then joins two HL hemimolecules to form a single complete molecule. So H2 and L2 dimers can also associate to form the complete molecule, and light chains can be effectively secreted from the plasma cells, unlike for the free heavy chains, which are synthesized and transported to the endoplasmic reticulum, where they are glycosylated. So for immunoglobulin M and A, they have one additional polypeptide chain called the J chain. It is very important for you to remember this, the J chain. It is found in immunoglobulin M and J and A. And what is the function of this J chain? So the J chain has some role in initiating polymerization. So we have to discuss the 5 classes of immunoglobulins that we have already highlighted. So and these 5 classes of immunoglobulins are achieved based on their differences in their heavy chains. So we have immunoglobulin G, A, M, E, and D, and you can remember them using the acronym GAMED. So starting with immunoglobulin G, so this one is composed of 2 heavy chains and 2 light chains, and the bonds are linked by disulfide bonds. So this immunoglobulin is divalent and contains 2 identical antigen binding sites. And we have 4 subclasses of immunoglobulin G, that is subclass 1, subclass 2, subclass 3, and subclass 4. And out of the 4 subclasses, immunoglobulin G1 is the most dominant. So immunoglobulin G1 and immunoglobulin G3 are more effective opsonizers compared to their counterparts, that is immunoglobulin G2 and G4. So This type of immunoglobulin is the only immunoglobulin that can cross the placenta, and this happens when its Fc portion binds to the FcRn receptor on the surface of the placental cells. So the FcRn receptors can transport the maternal immunoglobulin across the placenta into the fetal circulation. And because we are saying this immunoglobulin can cross the placenta, so it is the most abundant immunoglobulin in newborns. So it can activate the complement, and it is important for you to know that only immunoglobulin M and G can activate complement. They also act as opsonizers to enhance phagocytosis, and the surface of the phagocyte contains receptors for the heavy chain of immunoglobulin G. Moving on to immunoglobulin A, so this one is the main immunoglobulin found in secretions, for example, saliva, the tears, respiratory tract secretions, intestinal tract secretions, the genital tract secretions, and also for colostrum. And secretory immunoglobulin A are dimers composed of 2 heavy chains and 2 light chains units, plus 1 unit of J chain and secretory components. So serum immunoglobulin A are monomers composed of single H2L2 units. So here we can see the secretory component. So this is where we have the secretory component, and it is important for you to know the function of the secretory component. So secretory component is a polypeptide made by epithelial cells, and it helps in passage of immunoglobulin A to the mucosal surface and prevents its degradation by proteolytic enzyme in the intestinal tract. So that is very important for you to remember. So next we will look at mechanisms of transport of immunoglobulin A molecule across an epithelial cell. And this one is very important for you to understand how it happens. So how does this happen? So immunoglobulin A molecule binds to a transmembrane receptor protein on the surface of a secretory epithelial cell. So we can see that binding taking place at this region here. So you have the immunoglobulin A binding to the secretory transmembrane receptor. Now what follows next? So the receptor immunoglobulin A complexes are ingested by receptor mediated endocytosis, transferred across the epithelial cell cytoplasm in vesicles, and secreted into the lumen on the opposite side of the cell by exocytosis. So when exposed to the lumen, the part of the Fc receptor protein that is bound to the immunoglobulin A dimer is cleaved from its transmembrane tail, thereby releasing the antibody. So that is how that immunoglobulin is transported across an epithelial cell. Moving on to immunoglobulin M, so this is the main immunoglobulin that mediates primary immune response. So it is found as monomers on the surface of all B lymphocytes, where it acts as antigen binding receptor. So it is found as pentamer in serum composed of 5 H2L2 units plus 1 J chain. So immunoglobulin M pentamer has 10 antigen binding sites, and we have already said that this type of immunoglobulin can fix complement. Moving on to immunoglobulin E, so it can mediate hypersensitivity reaction, and it is the main defense against worm infections and also sometimes referred to as helminth infections. So the reason for this is because worms are too large for phagocytes to ingest, so they can be killed by worm destroying enzymes released by eosinophils. So this type of immunoglobulin which are specific for worm protein binds to the receptors on the surface of eosinophils and trigger antibody dependent cellular cytotoxicity, which then will end up killing the worms. Lastly, we shall look at immunoglobulin D. So this one is an immunoglobulin that has got no known function, and it is found on the surface of many B lymphocytes, and they participate in as antigen receptors, or they may act as antigen receptor for that matter. So what are the general functions of antibodies? So the general functions of antibodies include: number one, they neutralize toxins and viruses. They facilitate phagocytosis by opsonization of microbes. They activate complement, leading to complement-mediated activities against microbes. They prevent attachment of microbes to mucosal surface, and they can also have catalytic functions. So we have a table here that gives a summary of the antibody and their respective functions. So for immunoglobulin G, they act as opsonizers, and they also fix complement. They also neutralize bacteria, toxins, and viruses. And we have said this is the only type of immunoglobulin that can cross the placenta. So for immunoglobulin A, this is the secretory immunoglobulin that prevents attachment of bacteria and viruses to mucosal membrane. Moving on to immunoglobulin M, so this one fixes complement, and it also acts as antigen receptor of surface of B lymphocytes. For immunoglobulin E, it mediates immediate hypersensitivity, that is type 1, type 2, and type 3, and it also defends the host against worm infections. And the function of immunoglobulin D is unknown or uncertain. Moving on to polyclonal and monoclonal antibodies. So poly means several or many. So this type of antibodies are produced by several different clones on the plasma cells in response to typical antigens, and they are heterogeneous. Whereas for monoclonal antibodies, mono means one, so they are formed by single clones of plasma cells, and they are heterogeneous. For example, myeloma is a classical example of plasma cell tumors that are used to produce monoclonal antibodies. Next, we shall look at the concept of isotypes, allotypes, and idiotypes. So isotypes refers to the particular constant region of the light or heavy chain of the immunoglobulin. So all isotypes are found in all normal humans. For allotypes, these are additional antigenic features of immunoglobulins that differ among individuals. And idiotypes are antigenic determinants formed by specific amino acids in hypervariable regions. Then lastly, we shall look at metabolism of immunoglobulins. It's important for you to know that the half-life of immunoglobulins is used to assess the catabolic rate of the immunoglobulins. And half-life corresponds to the time taken for a reduction to a half of a circulating immunoglobulin concentration after equilibrium has been reached. So this is usually determined by injecting an immunoglobulin labeled with a radioisotope. For example, we are having this immunoglobulin that has been labeled with a radioisotope here. So for example, when we consider immunoglobulin G, so it is the immunoglobulin class with the longest half-life of an average of about 21 days, with the exception of the subclass 3. Understand, we said immunoglobulin G has 4 subclasses. So all of them will have an average of the half-life of about 21 days, except for the immunoglobulin G3. So for G3, it is the one that has the shorter half-life. That is of an average of about 7 days. Whereas for immunoglobulin A, it will have the half-life of about 5 to 6 days. And for immunoglobulin M, it will have the half-life of about 5 days. So this concludes our third presentation on immunoglobulins. And in case someone finds this helpful, I will really appreciate your comment, and you can share the video. So next we shall discuss on immunoglobulin genes. And if you want to know more on the structure and the function of immunoglobulins, I will recommend you confirm from your textbooks. So until next time, be blessed and keep safe. Thank you.